For the third postulate, the disease must be reproduced when a pure culture is introduced into a non-disease susceptible host. The molecular Koch postulate is that reversion or replacement of the mutated gene with the wild-type gene should lead to restoration of pathogenicity or virulence. And the molecular guidelines for that is that the copy number of a pathogen-associated nucleic acid sequence should decrease or become undetectable with resolution of the disease and should increase with relapse or recurrence of disease. The fourth postulate, the microbe must be recoverable from an experimentally infected host. And there are three molecular guidelines for that. There are, first, the presence of pathogen-associated nucleic acid sequence in healthy subjects should help predict the subsequent development disease. Diseased tissue should have higher copy number than normal tissue. And the last one is that this sequence-based findings should be reproducible. When we talk about the infectious process, then we talk about the entry of the microbe into the human being, into the body, and then colonization of the microbes, addition and invasion into the cells, and then the pathogenic action. In the first slide, we have talked about the three main components of the disease. They are host, agent, and environment. This slide shows you how the correlation between the microbe or the agent and the host. There are three components of the microbe which influence whether the microbes will produce the disease or not. First is the virulence factor, the percentage of optimal infectious dose, and the correct portal of entry. The virulence, of course, should be high. The percentage of the infectious dose should be optimal. And to produce the disease, then the microbes should enter the body through the correct portal. And from the host, there are three components as well. They are the genetic profile that resists the microbe. This is the component of the non-specific defenses. And then previous exposure to microbe. This is the specific immunity. And the third one is the general level of health, like the nutrition, and is there any underlying disease or other things. When there is a previous exposure to the microbe, means that the host has already had the antibody to the microbe. When there is no previous exposure, means that the host doesn't have any antibody to that particular microbe. Considering these two main components, there are three possible outcomes. So when, for example, when the microbe has high virulence, optimal infectious dose, but its entry through the incorrect portal, then it will not produce any disease, and the microbe will pass through unnoticed, and it will be excreted from the body. The second outcome is the microbe become established without disease. This outcome can be appeared when the microbe has the virulency, entry the body through the right portal with the optimal dose, but the host has already had the specific immunity to this microbe, then the disease will not be produced. The third outcome is the microbe causes disease, and this will be appeared when the microbe has the virulence and entry the body through the correct portal with the optimum dose, and the host has not had the antibody yet. There are three main portals of entry: skin, mucous membrane, and placenta. As long as our skin is intact, then there is no microbes will enter the body. But if there is a microlesion, then it is a very good entry of the microbes to enter the body and produce diseases. The second portal of entry is mucous membrane. We know that all of the tracts in our body are surfaced by the mucous membrane, and conjunctiva is also the thin membrane covering the surface of the eyeball and the underside of each eyelid. As long as the mucous membrane is intact, it is also no microbes enter the body. When there is a microlesion, the microbe will enter through this. The third portal of entry is placenta. There is a barrier, but some of pathogen can break the barrier and enter the fetus to produce diseases. There is a parenteral route. It is actually not a portal entry, but instead a means by which the portals of entry can be circumvented. Once the microbes enter the body, they will try to colonize the surface of the body, and to colonize a site populated by normal flora, it is of course not an easy thing. The new arrival must compete successfully with established organisms for space and nutrients, overcome their toxic product, and counter the body's defenses aimed at protecting the surfaces, which is IgA proteases. When the microbes successfully colonize the surface of the body, then they will attach themselves to cells. In this process, they need the adhesion factors. There are some adhesion factors we know. First is the specialized structures, which is belongs to protozoa. We call it adhesion disc. There is also suckers or hooks in helminths, and also ligands. Ligands is the surface lipoprotein and glycoprotein, and we can found ligands in bacteria or viruses. The ligand in bacteria is named adesin, found on fimbriae, flagella, and glycocalyces. The second adhesion factors is ligands, which can be found in bacteria and viruses. In bacteria, we call it as adesin, and in viruses, we call it as attachment protein. There are two types of bacterial adherence mechanisms. They are pili. This is the rod-shaped protein structure that extend from the bacterial surface and bind to host cell surface molecules. The second one is the afimbrial adhesins. This is the bacterial surface protein that are not organized in a rod-like structure and mediate tight binding between bacteria and host. There are some factors support the adherence mechanism. Pili and fimbriae will mediate the attachment, and the pili constantly lose and reform, and it allows some bacteria avoid host defenses. Adhesin protein and also signal transduction. Loss of ability to make ligands happen because of genetic change or mutation, or exposure to certain physical or chemical agent, and the loss will make the microbes harmless or avirulent. Some bacterial pathogens do not attach to host cells directly, but instead interact with each other to form a sticky web of bacteria and polysaccharides called a biofilm.